let's talk a little bit more about the lens. It's a biconvex item. It is designed to refract or bend light. Remember, we talked that when you pass light rays through clear medias of differing densities, the differing in the densities will actually bend the light. And with our spoon right here, oh, I did it again. With our spoon right here, we're showing that the spoon actually looks larger underwater because of the refractive power of water itself. So water is a different density from actual air, even the glass that it's going through, and it causes the light rays to actually bend to where we actually get a magnification of that image. And that's why we use oil with oil immersion. Because if you've noticed, when the objective for the oil immersion lens, when you compare how far it is from the slide, it's about the same distance, not much difference. And the only real difference is actually that drop of oil that is actually placed on the object is actually used to actually magnify it. So curved surfaces are what we're moving into next, a little bit more about lenses. Transparent object curved on one or both sides, and whenever we think about this, we're actually thinking about glasses, for example. Convex, remember if you're vexed because your belly's protruding. Curved outward. Concave, you're actually happy because your belly is actually flat and actually curved inward. What happens with a convex surface, it will actually cause light to converge. So the light rays will bend towards one another is what that's telling you, coming off of various areas of an object. So if convex causes light to converge, what do you think concave does? Diverge. And exactly that. So light diverges. And it's actually due to how it hits the surface. So let's do a little picture off to the side here. Here is a convex, sorry about that. And now here's concave. And so when light is coming in, when it's right in the middle, it'll pass straight on through. But if it's hitting it off to the side, like that, it causes the light ray to actually be deflected kind of in a 90-degree fashion. So that's converging. Now if we follow this same principle and we're hitting straight in the middle, it'll go straight through. But if we're hitting, the light rays are actually hitting off to the side and we follow it, it will actually diverge. So our lens, based upon its shape, will affect how we bend the light rays. We're going to talk now about focusing. That's the main topic here, focusing, and focusing for things in the distance or things up close, near vision. Accommodation is listed up here first, and it is indeed performed by the ciliary body, and it will be associated with near vision. So accommodation is a term that we will actually use with near vision, and this is due to contraction of the ciliary body. And we're going to look at this first, then we'll go into distant vision and more about near vision. Take a look at this picture right here, and I want you to realize that we have taken the eyeball and we have made a frontal or coronal incision through it, and now we are looking at the first half of that eyeball from behind, in other words. So you easily see the lens right here in the middle. You easily see the suspensory ligaments or ciliary zonules, and you see the ciliary muscle of the ciliary body. Now, when you take a look at those fascicles, that's the proper term, but we like to say fibers or the grain of this muscle, how would you describe that to me? How would you say that the arrangement of these fibers or fascicles are to you when you take a look at that picture? Circumferential? Yes. Circular? Are they radial? Don't look radial to me at all. They look circular or circumferential. So when that muscle contracts, what will it do? Will it get closer to the lens or further away from the lens? It'll get closer to the lens. If it gets closer to the lens, because that's what I'm talking about, accommodation is contraction of the ciliary muscle of the ciliary body. If it gets closer to the lens, what happens to the tensions on the suspensory ligaments? Decreases. So this is very important that you understand that the ciliary body is, that the muscles are circumferentially arranged on the ciliary body. So when that muscle contracts, that's going to get closer to the lens, and that's going to cause a decrease in tension on the lens capsule and the lens itself. Well, what will that do to the thickness of the lens? Will it make it thicker or thinner? Thicker. Okay, good. This is important. If you get this concept, you are in great shape. So, associated with near vision for accommodation due to contraction of the ciliary body. So when you are accommodating, your ciliary body is contracting and the tension on the suspensory ligaments or ciliary zonules decreases and the lens thickens. So all of these are a natural part of accommodation. With that in mind, let's take a look now at distant vision. And near vision. What you want to do is you want to change the shape of the lens to allow the light rays to converge on the macula lutea, fovea centralis. Think of the macula lutea as the sweet spot. We do get obviously light rays landing in other areas, but to see the best, you want it on the sweet spot, the macula lutea and the fovea centralis, which is straight back. So in order to see those things, whether it's far away or close, we need that lens to change its shape to ensure that the light rays are bent or not in order to get the light rays to that sweet spot. When things are far away and you're looking straight at it far away, is it big or is it small, the item that you're looking at, realistically? It's small. So if you're looking at something in the distance, you're focusing on some animal in the distance, the animal appears smaller than if the animal's up closer to you, right? Okay, so do you need to really bend those light rays much that are coming from that animal that's far away, that's in a very small area? Do you need to bend those much? So do you want your lens thin or do you want your lens fat, excuse me? Fat or thin? Thin. And so that's what we do. When we are looking at things in the distance, our ciliary muscle is relaxed, which means what about the tension on the suspensory ligaments? They are increased, which means the lens is flatter. So all of those things you have to keep in mind. You want everything to go to the sweet spot, so we want to bend the light rays coming in to help maximize their ability to hit that nice little macula lutea and fovea centralis sweet spot. So ciliary muscles are relaxed. This means lens flatter, not fatter, but flatter or thinner. And tension on zonules is high. So you can bet that the opposite's going to happen now for when we're looking at things up close. When we're looking at things up close, the item is big, is it not? But we want to shrink it down to where those rays coming in will hit the sweet spot. We don't want all those rays hitting all over the retina. We want them to go to the sweet spot. So we need to bend them more. And when we bend them more, the lens is always thicker. So for close vision, what we will have here is the ciliary muscle is contracted. So it's the opposite of the other. Tension on zonules is relaxed. This causes the lens to thicken or get fatter. And we'll have better refraction or bending of the light to converge. Near point, that is a terminology that's associated with how close can you put your hand to your face and see all of the lovely fingerprint ridges and creases and everything clearly. That's your near point, the closest point of focus. This depends upon how fat the lens can become. So when you're looking at your hand and you're looking at the various tracings of your lifelines and things like that, you want to be able to see them really clearly. And when you bring your hand in further than, closer than that, things become fuzzy. Okay, so the near point's that area where you can take your hand close to your face and then bring it on out, and that nice, good focus is your near point. As you age, your near point increases. So the near point is measured from your eye to the object. So what does this mean about your near point? Does your hand get closer to your face or further from your face? Further from your face. So that's what it means. You have to make sure that you're following the terminology well. Near point increases. So the distance that you can see an object clearly as you age gets further away from your face. What is that telling you about the lens itself? Is it able to get nice and juicy plump as well as it did when you were younger? No. So what's happening here, and that's why in our notes it says loss of elasticity with age. So the elasticity is, realize that if we are relaxed, suspensory ciliary body is relaxed, tension on the zonules is increased, and lens is flat. Then when we want to look at things up close, we contract the ciliary body, and the tension on the zonules gets less, and so then our lens is able to go bloop on out. Well, what do you guys know about elasticity of your clothing over time? The waistbands in your favorite pair of lounging pants or sweatpants. What do you know about an elastic waistband over time? It loses its elasticity. And so that's the very same phenomenon that we're experiencing. We're experiencing degradation of that elastic nature over time. And so that's why as you age, this happens. Now, another thing to consider is some people never seem to actually have problems with their near point getting further away from their face. So they're still able to read up close. And so what can you do that will minimize oxidative damage due to photodegradation? Do you guys know what I'm trying to say when I say photodegradation? Put a piece of clothing out in the sun and leave it there for years at a time. Then pick it up and look at the other side. Which is darker? The other side. So photodegradation, because isn't that what our lens is dealing with all the time? What can you do to help minimize photodegradation? Sunglasses. That's right, sunglasses. So indeed, wearing sunglasses is helpful for you to minimize the photodegradation that will occur to your lens over time. And so with your little children outside and stuff like that playing, it's really hard to get them to wear sunglasses, isn't it? They're too busy playing to put something on their face. But that's something to consider. The amount of light exposure, sunlight exposure particularly, is going to contribute to what I call normal photodegradation to any object that it encounters. So the elasticity is lost with age. Near point increases, which means your arms aren't long enough. Because when it first starts to happen, you just move your arms or your hands further away from your face. But after a while, they aren't long enough, and then you have to wear these little glasses called cheaters in order to actually see. You're using a different lens to help bend the light so that you can actually see things up close. Let me ask you, so this is all due to photo degradation. Over time. If your lens becomes less elastic, what does that tell you about the nutrient's ability to actually get to the center? It decreases, and that's another reason why, as we age, the cataract phenomenon seems to be more prevalent in individuals. So cataracts are also a result of loss of elasticity and nutrient supply to the center of a lens, for example. So near point, what is happening too with the ciliary body? When you look at things up close, it's constricting, and so that's a form of accommodation. Constriction of the pupils also occurs. So that was constriction of the ciliary body. Now we're going to go to constriction of the pupil. Enhances the effect of accommodation. It allows you, when your pupil's really big, a lot of information goes in and a lot of bending has to occur, right? Well, if your pupil gets smaller, you're putting in less information, correct? And so your bending doesn't have to always be so extreme. So constriction of the pupils indeed enhances the effect of accommodation. So you are able to focus on just that which is in front of you. So you have a constricted pupil and you have a fat lens. Are these the results of sympathetic or parasympathetic innervation? Because remember, this is something you don't have any control over. Parasympathetic, rest, digest. You're getting ready to have dinner, folks. So you're just focusing on that which is close to you. Convergence of the eyes also occurs during this process of accommodation, things that you're looking at up close, correct? That is controlled by the oculomotor nerve. And so what muscles are you going to contract to allow your eyeballs to converge? It's a rectus muscle, correct. Which rectus muscle? Medial rectus, both or one or the other? Both, that's right. So controlled by the oculomotor nerve, and so we will contract our medial recti. Recti, remember, is plural. Rectus would be singular. So convergence, contract, and indeed required for close vision. Let's talk about a few visual problems. We have 3 problems listed here, and one is not a problem. The not problem is the term emetropia. Emetropia is normal vision, and what number is that typically when you measure it with the Snelling eye chart? 20/20, and if you're lucky, 20/15. So it's how far, 20/20 is that what most people see clearly at 20 feet, you also see it clearly at 20 feet. Also, 20/15 is what most people see clearly at 15 feet. You also see it very clearly at 20 feet. So it's a good rule of thumb, and most people can see far away without any difficulty with that. Two terms here that are related to how far an object is from you to see clearly is myopia and hyperopia. Myopia is called nearsightedness. Can see up close clearly. So you can see things up close. Myopia, nearsightedness. So hyperopia or farsightedness means you can see things clearly in the distance. It's always nice to actually work with these as pairs. Get one memorized and you got the other one memorized well, or just work on the conceptual thinking there. We have some pictures down below. Let's talk about emmetropia first, normal vision. So remember, we are trying to get everything in the sweet spot on the retina. So we need those light rays to converge on the retina in order to see things, right? To see things clearly. So that's what this is talking about with the focal plane or focal point. And to me, I more or less call it a focal point because it's all converging down to this one nice area to see well. When you have myopia or nearsightedness, let's go on over to here. What happens is the focal point is in front of the retina. It's not on the retina, it's in front of the retina. And this process is due to a problem with the length of the eyeball. So in order for the focal point, for everything else to be normal, and the focal point to be in front of the retina, the eyeball has to be too long. So that's what we see here in our notes. Eyeball too long. So distance objects appear blurry. And so what you try to do is you try to change the focal point so it actually goes further back. So instead of converging so soon, you want the rays to actually diverge a little bit before they come on into the eye. So that's myopia. Hyperopia is the opposite of myopia. When hyperopia, your focal point, if you notice here, is behind the eye. Well, the most common situation in which the focal point would be behind the eye would be if the eyeball was too short. So people who are farsighted have short eyeballs, and people who are nearsighted have long eyeballs. And it's really true. It really is that way. It's the actual length of the eyeballs. So can see up close with myopia, distant objects, focused in front of retina. Not on the retina, but in front of the retina. Coming together too soon. Go ahead and write it in here. It's in the notes, but I'll write eyeball too long. Hyperopia. We have our near objects being focused behind the retina. And this is the eyeball too short. There is a surgical procedure that you can do for people who have myopia. Do you guys know what it is to help correct their myopia? LASIK surgery. Anybody ever heard of the really old one that actually first occurred before LASIK, which was radial keratotomy? So radial keratotomy, take the cornea and go around the edge of the cornea. Let me show you. And make little cuts into that cornea. What will those cuts do whenever they heal? Will they scar? Yeah. And when they scar, what do a lot of scars do? They pull. They create traction on things, correct? And so radial keratotomy is actually where you actually make radial incisions into the cornea, creating scar tissue to what to the cornea? Make it bulge more or make it flatten? And so that's one of the maneuvers to do that, because if you make it flatten, then you will be able to cause those light rays to converge more on the retina. So that's radial keratotomy. Tommy. Now LASIX. Oh, how do you spell LASIX? L-A-S-I-X? LASIX. Oh, that's not it. Is it LASIK with a K? Yeah, LASIK. LASIX is the it's a diuretic that you give people. So, I hate when they actually come up with names that are very similar to what you know out there. With LASIK, what you do is you take a look at somebody's corneas, because they do bulge from the sclera, and you go in with a knife and you cut off a section of their cornea. When you cut off the section of a cornea, we are not protruding as much anymore, correct? You literally make a wedge section out. You open it up and then cut out a section below this superior cut here. And then you put this right back on. And this effectively decreases the height of the hill, correct? Which is decreasing the bulge also. So we want to, in either procedure, we decrease the bulge, B-U-L-D-G, of the cornea. Or in other words, flatten it, I guess is a nicer way of saying it. And when you flatten it, you bend the light less. Our cornea is the major item that does the most refraction or bending of light. But then when we want to actually see things up close, we have within our eye this pliable lens that can get fatter or thinner based upon what distance the object is from our eye. So that's a little bit about those procedures. Hyperopia, there's that right there. And it shows you the various lenses that will be used. Now let me ask you a question. Let's say the eyeball is a normal size, like emmetropia, but we are focusing in front of the retina or before the retina. What's wrong with the lens then? Let's say that the lens is the problem. What's wrong with the lens that we would actually cause our light rays to converge too soon? Is the lens is too fat. So the eye is too long, or theoretically the lens could be too fat. And it's mostly the eye is too long. Very rarely is the lens ever too fat. With that in mind then, with hyperopia, and it's a normal size or length eyeball, and we're still focusing behind, what does that mean then to you there also? Lens is too thin or flat. So those are also potential things. And you'll see that in the old exams, because they actually talk about whether it's too fat or too flat, too long or too short. And so you have to remember how to pair it up and how to actually figure out the convergence in the focal point. Astigmatism, which is all written over with my arrows here, is due to unequal areas of either our cornea or our lens. So instead of being beautifully symmetrical, what they're telling you with astigmatism, there's a little bit of a defect in this one area. Or there's a little bit, we can make it a little bit more obvious. There's a little bit of a defect, and that defect will affect how the light rays are bent. And you can actually correct that with lenses, hard lenses, or contact lenses for that. That can be in either the cornea or the lens itself, causing the astigmatism, causing the light rays to actually hit the retina in an area that they should not. So that's easy stuff. Now we're coming to the complicated stuff, phototransduction. So this is the real physiology of vision. We are going to need to convert light energy into an electrical signal, which will go back and register in our visual cortex. Because everything that we experience, in order for us to know it, we have to experience in our cortex. You guys remember that? If we take a chunk of cortex out, will you ever know it happened? Even if it's happening? Our awareness is cortical. And so we have to get this information about the light that's hitting the retina actually to our visual cortex. And that's going to involve the process of turning light rays into the electrical signal, phototransduction. Rods and cones are modified neurons, and they look a bit like columnar cells, and they are a bit inverted. And their receptive tips are buried in the pigmented layer of the retina, not of the choroid, but of the retina. So as we take a look at this picture here, this is showing you a pigmented cell nucleus, and here is our rods and cones with their tips embedded into the pigmented layer of the retina. This is important because we only want to stimulate the appropriate photoreceptor, and so this is going to help prevent light scatter further. Remember, our choroid helps to prevent light scatter, so does the pigmented layer of the retina. The outer segment is where light will actually interact with the photopigments. And this is the area, outer segment right here, that is actually buried in the pigmented layer. Retinol, which is a component of our photopigments, plus another component of our photopigment called opsins, form just 4 light-sensitive photopigments. So the richness of the color of your world is only dependent upon 4 different photopigments. Just like taste and just like smell, it's how much we stimulate them as to how rich the color variation is in our world. So retinol is going to absorb the light, and retinol has an opsin that it combines with to create the 4 distinct photopigments of the human eye. Retinol is made from vitamin A. Which is actually made by our pigmented cells of the retina. So in order for retinol to be made, it requires vitamin A, and it gets it from the pigmented cells, and it's right there embedded into the pigmented cells. So it's easy for it to get that vitamin A to actually make its retinol aspect of our photopigments. Our notes here say rods, more numerous than cones, primarily located within the peripheral retina, specialized for dim light or night vision. Rods are for low light levels. Rods do not help us in distinguishing color, and they do not give us clarity of the item that we are looking at. So rods function in low light. These guys are low light photoreceptors. Low light means not a lot of light. So they are actually active when there is not a lot of light. Cones, on the other hand, our notes say are least numerous in the rods and absolutely primarily located within the fovea centralis plus macula lutea. Respond to stimulation by bright light. So this is intense light or highlight levels. and they allow us to recognize color vision and give us sharpness of vision, which I refer to also as clarity. We have cones that are going to be for blue, green, and red. So what you see is the addition and subtraction of various photoreceptors in your appreciation for yellow and pink and all the myriad colors that we actually see and experience. Outer segment, pigmented layer, vitamin A, produced by pigmented layer, helps maintain the healthy photoreceptor area of our rods and cones. This guy here is a cone. This is showing us two rods. So it's not the absolute number of rods to cones, but it shows you we have more rods than cones. So that's the takeaway message from that. From the outer segment, we have this interesting area full of mitochondria. That's the inner segment. Then we come up to the cell body where you guys saw all the nuclei. Remember the retina, when you take a look at it histologically, it has this huge layer of nuclei. That's what we're actually able to see, not so well the outer and inner segment. And then we have the ends here that will synapse to bipolar cells. So rods and cones synapse to bipolars, and bipolars synapse to ganglions, and the axons from the ganglions take all that information to create the optic nerve, which will then travel to the visual cortex of the occipital lobes. Not too complicated yet. Notes. Rods, responsible for black and white, also referred to as our night vision. Their photopigment is rhodopsin. So we actually have retinol plus an opsin up here, it states. And for rods, it's retinol plus rhodopsin. Again, sensitive to low light levels. So rhodopsin is sensitive to low light levels. Very little light stimulates rods. Stimulating rods is activating rods. When you stimulate the rods or activate the rods, this causes the breakdown of their photopigment rhodopsin. Activating, stimulating, breaking down rhodopsin. Location. Highest concentration of our rods, as noted earlier, is at the periphery of the retina, and as you come closer to the macula lutea, you'll find that there's more cones interspersed in that area. But then you still have some rods in the macula lutea. You have no rods in the fovea centralis. So this is consistent with this. None in fovea. A few in macula lutea. The reason you can't see well in low light is because of the number of rods giving information to one ganglion cell. Our notes here say low visual acuity means poor clarity, not crisp edges. And what we have here is we actually have 100 or more rods will actually finally synapse with a ganglion cell. So let's just use the number 100. So 100 rods, we'll say, will synapse with a few bipolars, which will synapse only with a ganglion cell. So what this is telling you, we have a large area, and any spot in this area that gets activated will activate a ganglion cell ultimately with rods. On the other hand, with cones, we'll come on down here with cones, we have one cone feeding into a single ganglion cell. So we'll say ganglion cell off to the side, and cone, and it's just one cone to one ganglion cell. We have many potential rods to one ganglion cell. Which gives you more precise information, a one-to-one connection or this zone-to-one connection? One-to-one connection. And so that's another reason why our clarity is not so good with the land of rods, because the land of rods don't have that one-to-one connection. They have, oh, over here and over here and over here, I stimulate this one ganglion cell. Whereas a cone says, I'm going to stimulate this one, the one next to me is going to stimulate the next one, the one next to me is going to stimulate the next one. So these cones are having a one-to-one connection, which means more discriminating information is being sent to the brain about what we're actually seeing or actually is stimulating the brain. So 100 or more feeding into each ganglion cell, low visual acuity. Big area to just one cell gives us less true info. Now, so far you guys I think can pretty much follow along with that. This is where we're getting into some areas that will make you a little confused. Rhodopsin in the dark. So that means no light, correct? Rhodopsin in the dark. Close your eyes. We're talking about that situation right now. Rhodopsin in the dark. Those rods are constantly releasing neurotransmitters. So when your eyes are closed, your rods are constantly releasing neurotransmitters. What do you see when your eyes are closed? Nothing, black. And if you're releasing neurotransmitter, the way you ordinarily think about it is what happens to the bipolar cells. You stimulate them, and then what happens to the ganglion cells? You stimulate that, and then what happens to your cortex? Should stimulate that. But that's because you are thinking only of EPSPs. You're only thinking of a neurotransmitter that gives you an excitatory postsynaptic potential in the bipolar cells. What if I tell you that the neurotransmitters that are being released by the rods continuously gives you IPSPs? Inhibits the bipolars from firing. Well, if the bipolars don't fire, the ganglions don't fire, and the visual cortex sees nothing. And that's what's different. You're so used to thinking of everything as being excitatory that it takes a big brain shift for you to think of it as being inhibitory. So it's important that you realize the NT released is an inhibitory neurotransmitter. And what's interesting, it actually causes the bipolars to hyperpolarize. You guys remember what's important about hyperpolarization of a neuron?
It takes more excitatory to come back.
Absolutely. It's harder to get them to fire. So we're releasing an inhibitory neurotransmitter on our bipolar cells, and we're making them hyperpolarized, which makes them more difficult to fire when we are in the dark. So now let's take a look at what happens when some light, a little bit, we don't need a lot, we just opened up our eyes and it's still pretty dusky outside. Rhodopsin is now being activated or stimulated because we're applying a low level of light to that. That means rods do not release this inhibitory neurotransmitter. And what does this tell you about the bipolars then? Are they be hyperpolarized anymore? No, the bipolars no longer hyperpolarized. The bipolars themselves are set to fire. And so that's why this inhibitory neurotransmitter is hard on them, because it stops them from firing. When you stop applying inhibitory neurotransmitter to your bipolars who want to fire, they will then fire. And so that's what happens with rhodopsin in the light. Rods no longer release this inhibitory neurotransmitter, and our bipolars then are able to fire. And you guys know fire means have an action potential or synapse actually across the light. It's important for you to realize light breaks down our opsins. Rods, this is also for our color vision, for our cones. Rods are slow to regenerate its opsin. Regen. And rods do not function well, or function at all, in high light situations. So a little bit of light will cause the opsin, the rhodopsin, to degrade, which will cause the rod to not fire its inhibitory neurotransmitter, which will activate our bipolars. A little bit of light. When you apply a whole bunch of light to the rods, it totally freaks them out. And as a matter of fact, that term is called bleaching. A whole bunch of light causes all those rods, not just a few rods, but all those rods to go, whoa, no longer functional, and it takes some time to regenerate their photopigment, which means that when you go from a very bright room into a dark tunnel, what do you know about your vision, your night vision?
Okay.
It's bad, right? Because rods are having a hard time coming back to what I call life. They're not dead, but they're not creating the rhodopsin at the rate that the cones can create their photopigment. So the rods are real slow to regenerate the rhodopsin to actually get back into functionality. So they've been bleached, and it takes a while for them to get back to becoming functional rods for you in the dark.
We had a sunspot, you know, I mean, 'cause that's not really rods, it's not function, it's a lighted situation, it's a lighted situation, we look the lighting of that spot, that bleaching as well.
Yeah, what happens is you still have, when you actually have that reverse image that you actually see, or somebody takes your picture with a flash, the big flash, and then suddenly you have that hole in your vision. Well, that actually hit your cones too. So the same process happens with your cones, but your cones are for clarity and color vision, and typically that's when that happens. And your cones also can be bleached, but their ability to regenerate their rhodopsins much faster, and so you get that color vision back so much faster. Rods do not function in high light situations. So going outside on a nice sunny day, we aren't using our rods, we are using, therefore, our cones. Okay, cones are responsible for color vision. We have 3 types of photopsins that respond to red, green, and blue light waves. They require a relatively high light intensity to stimulate. So stimulate means applying light to the receptive end of our cones. So when we stimulate them, our photopigment breaks down, but is regenerated quickly. Remember, with cones, it's the same phenomenon. When cones are stimulated, they fire. When cones are stimulated, then we are releasing oh, good Lord. Let me make sure I say it right. When rods are stimulated, when light hits them, they no longer release an inhibitory neurotransmitter. When cones, light hits them, they no longer release the inhibitory neurotransmitter. Rods and cones both release inhibitory neurotransmitter. So when you stimulate them, you essentially turn on the bipolars because you've stopped the release of the inhibitory neurotransmitter. Did I say that right? Do you guys understand the path that I'm trying to take? That's why this is a little bit more difficult. It's even difficult to try and say that and keep the details straight. So we need a lot of light to stimulate our cones. Photopigment breaks down, but is also regenerated quickly. And whenever we stim, we stop the release of inhibitory NT on bipolars, which allow them to fire, which will then allow the ganglion cells to fire, and the info goes to the cerebral cortex, which is in the occipital lobes. We have high visual acuity because one cone is going to send its precise information to a ganglion cell. And it is best vision when we look at object straight on in high light or good light situation. That's when we get our best visual acuity or clarity. If we want to see an object in dim light, will we try to look at it straight on? No, we want to actually not have it right in the center of our vision if we actually want to be able to see it best in dim light. So see object in dim light if not straight on. Okay, generating an image. Stimulate all photoreceptors. In other words, apply light to all of our photoreceptors, big time. All we see is white. So they're all being essentially bleached. at that stage, correct? All we see is white. All opsins are being broken down, and we see only white, which we can relate to bleach, which is what bleach does to your clothes, makes everything white. When you stimulate no photoreceptors, all we see is black, and this is due to continuous inhibitory neurotransmitter release. No information to the brain? Don't see anything. And that's interpreted as black. Light and dark adaptation. So that's what we already kind of touched on that topic. Light adaptation and dark adaptation. In light adaptation, what we're doing is we are going to go from a dark room into a light room. From dark into light. Getting used to the light. Light adaptation. From a dark room into a light room. Well, when we are in a dark, what do we know about degradation of the photopigments? Wait a second, I've got it wrong. Light adaptation. Nope, I got it right. Okay, when we're in the dark, what do we know about breakdown of our photopigments? Yeah, we're not breaking them down. So things are functioning properly. Breakdown of photopigments, not really happening to any degree. So rods and cones are fine, they're happy. Now we go into the light. What do we know about our rods when we go into a brightly lit room?
Yeah.
They're freaking out. Remember, rods are sensitive, and when you do anything to a sensitive person, they freak out, right? Okay, so rods are sensitive, cones are not. So we put rods in a brightly lit room. Rods lose ability to function. And that's our bleaching effect. So they do this temporarily as long as we're in a bright lit room. As long as we stay in a bright lit room, our rods are not really doing a great job. Because in a brightly lit room, we see colors really well, and we see the edges of the items that we're looking at very well, crisply. And so that's a function of the cones. So with cones, go into a light room. We know cones still work, but will have a moment of, depending upon the light level, of kind of freaking out. Have you guys ever gone outside where it's been so bright it hurts your eyes? That's what I'm talking about. It's so bright that even the cones got overstimulated, and so what do you typically want to do when it's so bright it hurts your eyes? Close them. So that's that photoreceptor reflex saying we've got too much light even for our cones to be happy with, and we're going to need to kind of squeak it on down to where we're not being so intensely overwhelmed. So cones still work, but we'll have a moment of overreactivity can cause you to close eyes somewhat. Because you're trying to actually let less light into the cones. So in this situation, you lose sensitivity, but gain acuity. Because sensitivity is for rods and acuity is for cones. You guys remember when we talked about taste, adaptation with taste? You know, that first lick of the ice cream is the best because you start to get used to it. The same thing happens actually with your cones. So the cones got this barrage of light and they kind of got a little freaked out with it, but still were able to function. And then they got used to the amount of light being applied and got a little toughened up. The rods don't have that ability. The cones actually have that ability to actually kind of change their sensitivity to light. And so what you will notice when you go from a dark to a bright room, visual acuity is best 5 to 10 minutes after you go into that light room or out into the lighted area. Your eyes don't hurt so much when you're out in that bright light. Dark adaptation is the opposite of light adaptation. So we're going from light into dark. Well, when you go into a light room, what do you know about rods right off the bat? They have been bleached. Rods are bleached. And so you go from a bleached rod situation into a situation where you need rods, correct? Because cones do not function in low light. So you're switching from a cone-only situation where the rods have been put off to the side, so to speak, with the bleaching, to now we're in a dark room. Cones don't even know how to function in that situation, and our rods have not been able to recreate their photopigment to where they actually can function properly. And so in order for the rods to actually create their photopigment to function properly and for you to actually see well in the dark, you need 20 to 30 minutes. To dark adapt so that you can have the rhodopsin reaccumulates. And the rods respond appropriately. This is something that you should probably go home tonight and review so that when we get together on Thursday, you can ask a few questions. Because you understand it right now because I'm holding your hand, walking you through this. But can you go home and explain this to somebody tonight? Yeah. So in order for you to really feel like you understand it after I talk you through it, you have to be able to talk somebody else through this. And it's not that easy. The last part of this portion of the chapter, and then we'll go into the ear for hearing and balance, is binocular vision and depth perception. Well, binocular vision means what? When you hear that word binocular? Two eyes. You notice it's linked here to this term called depth perception. So what about depth perception if you only have one eye? It's not any good. It ain't there. All you have, if you lose an eye as an adult, because some people do, they lose an eye from trauma as an adult, all you have for depth perception is what I call half memories. You have the one eye that's there, and when the other eye was there, the two work together. And so when the other one is gone, just like if you lost a spouse, you remember all the things you guys did together. And so when you activate that one eye, it remembers things based upon its experiences with the other eye. So you're still able to drive, but you're not always the best driver. Okay? So we do let adults who have lost their eye drive because of this memory the one eye has. But when it comes to really up close stuff, they're not very good. So it's always, you always should probably volunteer to drive for them if you want to feel safe. Yeah.
Contrasting that with a child that is, they don't have any depth perception, or then it's just a size comparison at that point.
It's a size comparison. If a child loses an eye, it becomes a size comparison. When you see something, an object further away versus up close, you learn what it looks like when it's up close or further away, type of thing. But if you've ever had, dogs, for example, and other animals that have their eyes off to the side, or even, well, cats too, when they have their eyes up front, if they lose an eye, what they'll start to do when they're looking at an object, in order to figure out where to jump, they'll shift. If you've ever noticed, they actually don't stay in one spot, they shift. So one eye, the other eye, one eye, the other eye. And that tells them how far the object is, and they're able to actually make those leaps of faith, so to speak. Well, let's go through the little bit of notes. Each eye, indeed, sees a slightly different image because you're at a slightly different angle, right? When you put your hand right in front of your nose and you look at, close one eye and you close the other, it definitely two different things that you're seeing with your hand. Same thing with the finger. If you take just a finger and you close one eye and look at it and close the other eye and look at it and it's straight in front of you, you're seeing slightly different things. So obviously depth perception is important for things up close, which is what we actually use it for. Optic chiasm. What's important for you to realize, the fibers from each eye cross over to the opposite cerebral hemisphere's visual cortex. And it is the medial half of the retina's information that crosses over. Let me draw, do you guys have pictures? For some reason, my picture did not come across when I did this. So I'm going to do a retina here, and I'm going to do a retina here. And I'm going to do my blind spot here, and my blind spot here. I'm going to go to my optic chiasm, and I'm going to cross on over to my optic tracts, and then I'm going to take and make a line to what should be my occipital lobes. The visual cortex. Now let's add some thickness to this. I had to create my first line. And so information from the lateral aspect on the right eye, so this will be my right, this will be my left, and the medial aspect on the left. Let's go ahead and follow that. So lateral aspect is going to go and, let's see here. Yep. And synapse here, and the medial aspect is going to go and synapse here. I'll do the other side. Left eye, lateral aspect is going to go on out and synapse on its side of the brain. And then the medial aspect is going to cross over and synapse on the opposite side of the brain. This is another reason why we're able to actually deal with a blind spot. If your object is right here, this object is going to have information hit the retina in various areas in that eye. And it's going to, because that's like the blind spot right there. Let's actually not do that. Spot. That won't. Let's have it hit this guy right there. This information from this guy is also going to hit this retina over that way, based upon where the pupil is located. And so we'll get information about this guy here and here. We'll be able to see it in both eyes. If we are right in the midline, so to speak, that information is going to hit here and hit here. And so that information from this guy is going to come to this spot here and this spot here. So things that are in the center are going to have information from two different eyes going to the same part of the brain. This is another reason why with the blind spot, if for some reason you're hitting the blind spot in one eye, you're actually hitting an area where photoreceptors are located in the other eye. And so you take that information and you fill in the defects that you are not getting from the other eye. And that's why you can actually have a blind spot and still function well, because your brain knows what should be there. So we'll just do those two examples. The third is actually also in your notes, so you can actually take a look at that. So medial half of the retina is going to cross over and go to the opposite side of the brain. And with depth perception, which is also referred to as 3D vision, because when they make 3D movies, they use two cameras and they sit the cameras side by side, which is like your eyes side by side. And when you use depth perception, everybody knows the distance between your eyes does not change, correct? But the object coming at you is changing how far it is from your eye. And as it comes to you, the closer it gets to you, the more medially rotated your eyes are, correct? And the more medially rotated your eyes are, that means the brain is telling you to contract the muscle more. And the more information to contract the muscle tells you that it's closer to you. And so this is the way your brain actually calculates the angle or how far away something is from you. If you're contracting your medial rectus a lot, does that tell your brain it's close to you or not so close? If you're contracting it only a little, further away. And so the amount of contraction of your medial recti is also providing information about how close or how far away the object is. So we'll stop there. I'll fill that in so that I can give you guys a clicker question.